Hello students, in today's class we are going to learn about methanogenesis. Methanogenesis is basically biological production of methane by the process which is known as methanogenesis. It is carried out by a group of archaebacteria which are known as methanogens, and the process involves the reduction of carbon dioxide by hydrogen to form methane, which is the major end product of methanogenesis process. There are also other minor end products which we will not be discussing here. So during the methanogenesis process, two categories of cofactors are required. First are known as the C1 carriers, and the second are known as those that will provide the electrons. So those that are known as the C1 carriers, they are the ones that are bonded with the different forms of intermediates that are formed during the reduction of carbon dioxide to methane, while the second category, which are known as the redox coenzymes, they will provide the required electrons for the reduction process. So here is the example of the first C1 carrier that is involved in the methanogenesis process, which is known as methanofuran. So it is involved in the first step of methanogenesis and acts as a C1 carrier. It binds with, so it basically has an amino nitrogen which is involved in bond formation with the reduced form of carbon dioxide to the first level. Methanopterin is involved in the intermediate steps of the reduction from carbon dioxide to methane, and it contains again an NH group. The nitrogen of the NH group is involved in bond formation in case of methanopterin C1 carrier. The third carrier that is involved is coenzyme M, and it contains an SH group through which it is able to act as a C1 carrier during the last step of methanogenesis, that is conversion of methyl group to ammonia. So here F430, it is a nickel 2 plus ion containing tetrapyrrole coenzyme, and it also acts as a part of the terminal methyl reductase enzyme complex. Then the second category of coenzymes are those that act as electron donors in methanogenesis process. So there are two types of electron donors. First is coenzyme F420, and it can switch from oxidized to reduced form by accepting protons and electrons. And in this way, it switches from oxidized to reduced state during the methanogenesis process. The second type of coenzyme is coenzyme B, which also contains an SH group, and the SH groups can be again, they can donate the hydrogen, and in the process they get oxidized during the reduction process. So now coming to the main methanogenesis pathway. The methanogenesis pathway can be divided into three stages. The initial stage, okay, this represents the initial stage, and that involves conversion of carbon dioxide to formyl group. Then we have the intermediate stages that involves reduction of formyl group to the methyl group, and then we have the third stage, that is the terminal stage, where the methyl group is further reduced to the end product, that is methane. So during the first step, that is conversion of carbon dioxide to the formyl group, pyridoxine or flavodoxine molecules, they provide the required electrons. They switch from oxidized to reduced form, and they provide the required electrons for the reduction. During this process, a molecule of water is removed, and the reduced form of carbon dioxide, that is the formyl group, is bonded to the first carrier, that is methanofuran. Here, the amino nitrogen will act as the molecule that is bonding with the formyl group. Now, after this, there is entry of the second carrier, that is methanopterin, which has an NH group. So the nitrogen of the NH group is involved in the bond formation. So at the beginning of the intermediate stage, the formyl group is transferred from methanofuran molecule to methanopterin molecule. And then there is a two-step reduction process during which F420 is going to provide the required electrons and protons for the reduction process, first to the methylene level and then to the methyl level. After reduction to the methyl level, there is entry of coenzyme M, which will act as carrier during the last step, that is the C1 carrier, and hence the methyl group is transferred from methanopterin to coenzyme M. So here the S sulfhydryl group, the sulfur of the sulfhydryl group is involved in the bond formation with the methyl group. And now during the last step, the enzyme methyl reductase complex, which consists of coenzyme M and F430, is involved in the reduction, that is conversion of the methyl group to methane group. So here coenzyme B, reduced form of coenzyme B, is going to provide the required electrons for the reduction. So this results in generation of methane, and in this process, coenzyme M and coenzyme B, they bond with each other through their respective sulfur groups, resulting in formation of a heterosulfide species. This slide explains about how energy generation will be linked with methanogenesis process. So as we just discussed, that the heterosulfide, which is a bond between coenzyme M and coenzyme B, is formed during the reduction process. And to regenerate, or the process to be continued, we need to regenerate the reduced form of coenzyme M and coenzyme B. And the pathway that leads to this regeneration is linked with ATP generation. So let us look at the ATP generation pathway. So here again, F420 can switch from oxidized to reduced form, and reduced form of F420 will provide the required protons and electrons to methanophenazine, which is the another molecule that is acting as an electron carrier, or you can say a redox electron carrier. So it will accept both protons and electrons from F420 and transfer only the electrons to cytochrome B, while the protons are excluded across the membrane. This generates a concentration of H plus ions across the outside of the membrane. While the electrons are transferred from cytochrome B to the enzyme heterosulfide reductase, which is involved in converting the heterosulfide, that is the bonded form of coenzyme B and M, into their respective reduced form. So for this, H plus ions are required, which are obtained from splitting up of the water molecule into H plus and OH minus ions. The H plus ions are used up during this reduction process, while the electrons that are required are obtained through the cytochrome B, through the electron transport chain. And the resulting OH minus ions will accumulate on the inner side of the membrane. So H plus ions across the outside and the OH minus ions across the inner side will contribute to generation of a proton motive force that is utilized by enzyme ATP synthetase to generate ATP molecules when it is shuffling back the H plus ions across the membrane. So the same things are written here for your reference. So thank you very much. That is the about methanogenesis. I request you to kindly like, share, and subscribe the video if you like the contents. Also, subscription will help you to directly browse through the website and gain access to all the videos. Thank you.